Hello everyone, my name is Kseniya Sholokhova and today I would like to present our project which is called A Search of Embryonic Selection Traces, Transmission Disequilibrium of Slightly Deleterious Variants from Parents to the Offspring with Down Syndrome. It is known that Down syndrome fetuses are subjected to high miscarriage incidence. However, the reason of this strong embryonic selection remains unknown. Why some fetuses are live-born, whereas others are not? This is the main question we are trying to answer. A potential way to find embryonic selection traces is to examine how slightly deleterious variants are transmitted from parents to the offspring. Now, please pay attention to the first picture marked as transmission deviations in healthy trias. Within our project, we worked with trias. Trio is a specific type of family that consists of three members, such as parents and a child. In case of Down syndrome, each trio consists of two parents without any chromosomal abnormalities and the child with Down syndrome. What we see in this picture is two cups filled with blue balls. A cup here symbolizes a human genome and blue balls symbolizing slightly deleterious variants. Some people have more slightly deleterious variants in their genome than others. From each parent, it is possible to obtain a huge variety of children's genotypes. You can see this distribution in the diagram. On average, it is expected that the total number of slightly deleterious variants in a child will be equal to the average mid-parent number of those variants. However, it is not always like that. Some alleles can be under or over-inherited from parents to the child, which will mark positive or negative deviation. But how? This can help us to find embryonic selection traces, right? According to the genetic handicap hypothesis, the total number of slightly deleterious variants can play a crucial role in the fetus viability. For further explanation, please pay attention to the second picture marked as transmission deviation in trias with Down syndrome. In the general case, if an individual has a genetic handicap, a very deleterious mutation, here it is a trisomy 21, then he or she should be alive only if the total load of slightly deleterious mutation is quite low. Otherwise, a miscarriage may happen. Accordingly, only well-adapted organisms that inherited from parents a small burden of slightly deleterious variants are able to tolerate the effects of extremely deleterious variant and survive. The figure depicts how the presence of extremely deleterious mutations such as trisomy can intensify embryonic selection by increasing the deviation of children from the expected mean. We interpret over-transmitted alleles as protection, which can partially compensate for the effect of trisomy, while we interpret under-transmitted alleles as aggravating, which enhance the effect of trisomy. And here we focused mostly on aggravation model hypothesis. So how did we calculate how the mutations are transmitted from the parents to offspring? We used a polygenic risk scores approach. Polygenic risk scores, or PRS, or genetic risks, approximate genome-wide predisposition to different complex diseases, which is transmitted from parents to the offspring. We calculated polygenic risk scores for both parents and compared average mid-parent score with the PRS of offspring. This comparison or contrast between mid-parent score and offspring score will reveal if genetic predisposition to a given phenotype randomly transmitted to a child or not, and any significant deviation of the contrast from zero will point out non-random transmission of the variance from parents to offspring and mark potential targets of embryonic selection. So this is what we did. We estimated polygenic risk scores, and for estimation we used Precise, which is polygenic risk score software, and Gene Atlas database, which contains a genetic association for 778 phenotypes, traits, and preliminary result on the figure 1 show that some traits demonstrated over or under inheritance from parents to the offspring, such as inflammatory diseases of the central nervous system, peripheral vascular disease, heart attack, myocardial infarction, and angina. Whether those deviations are random fluctuation or they mark the potential embryonic selection in Down syndrome trias is the question to address in the further analysis. Thank you for watching this video. If you have any questions, please do not hesitate to contact me. Here is my email. Have a nice day.